Good afternoon, Dr. Noor Isani Noor Jamil. My name is Tushitra Sri Armuga. My mention number 160978. I will be presenting on the topic of normal microbiota of the human skin and their roles. Firstly, what is human skin microbiota? Our skin is home to a vast community of microorganisms, collectively known as human skin microbiota. These organisms include bacteria, fungi, and viruses that reside on the surface and within the layer of our skin. The term normal microbiota refers to the beneficial microorganisms that coexist with us without causing harm. The normal microbiota of skin tend to inhibit transient microbe colonization by producing antibacterial substance and outcompeting other microbes that land on the surface of the skin. These help to protect the skin from pathogenic infection. Secondly, the diversity of the human skin microbiota. Human skin microbiota is incredibly diverse, with various species inhabiting different regions of the skin, especially in dry versus moist area. The figure on the left shows the major organisms commonly found in different locations of the healthy individual's skin and the external mucosa. Note that there is a significant variation among individuals. The availability of nutrition and moisture has an impact on the microbiota of a particular skin area. The microbiota of dry skin on the arms, legs, hands, and the feet differ from the moist skin in places like nostrils and underarms. Sebaceous gland density varies widely over the skin. In comparison to other body regions, the neck, nasal folds, and back all have fewer various microbial population in their sebaceous gland. Age, gender, environmental factors like humidity, temperature, and UV exposure, as well as lifestyle elements like nutrition, hygiene habits, and the antibiotic usage may all affect the composition of the microbiota. The microbiota on the skin can be classified into two groups. One is resident and the second is the transit. Resident microbiota is always present on the skin, while transient microbiota is temporarily and can be acquired through contact with other people or surfaces. Thirdly, the example of the human skin microbiota. Number 1. Bacteria, the commonly found on the skin, including Staphylococcus epidermidis, Propionibacterium acnes, and Corynebacterium species. These bacteria can be different in their abundance and distribution across the body. For example, sebaceous areas such as face tend to have a higher level of Propionibacterium species, while moist areas like armpit or groin have a distinct microbial community. Number 2. Fungi also form a part of the human skin microbiota. The most prevalent fungal genus found on the human skin is Malassezia, particularly Malassezia furfur. These fungi are particularly harmless and live in harmony with the skin. However, under certain conditions, they can cause skin disorders such as dandruff or seborrheic dermatitis. Lastly, viruses. Viruses are another component of the human skin microbiota, though their role is less understood compared to bacteria and fungi. Human papilloma viruses, HPV, and human herpes viruses, HHV, are examples of viruses that can be part of the human skin microbiota. Further research is needed to elucidate the significance of viral community in skin health. The last section is the role of the human skin microbiota. Number 1. Protection. The microbiota forms a physical barrier against pathogens, preventing their colonization and infection. For example, Staphylococcus epidermidis produce antimicrobial substances that help inhibit the growth of harmful bacteria, thus protecting the skin from potential infections. Number 2. The immune system regulation. Microbiota interaction with the immune system helps develop and maintain immune tolerance, preventing inflammatory responses and allergic. For example, Staphylococcus epidermis, Propionibacterium acnes, Malassezia species, and human papilloma viruses. Number 3. Nutrient competition. Microbes compete with potential harmful organisms for nutrients, limiting their growth and colonization. For example, Propionibacterium acnes utilize sebums, the oily substance produced by the sebaceous gland, as a nutrient source. By degrading sebum, it helps maintaining the balance of the lipid on the skin. Number 4. pH regulation. The microbiota helps to maintain the skin's natural pH by creating an inhospitable environment for pathogenic microorganisms such as Propionibacterium acnes produce fatty acid as metabolic byproducts which contribute to maintaining the acidic pH of the skin. This acidic environment inhibits the growth of the certain pathogenic bacteria. Lastly, skin barrier function. Microorganisms contribute to the integrity of the skin barriers, promoting hydration and preventing excessive moisture loss. For example, Malassezia species has the ability to break down skin lipids, particularly fatty acids derived from sebum. This contributes to the regulation of lipid balance on the skin. By metabolizing lipids, Malassezia helps maintain the integrity of the skin barriers and its water holding capacity, which is essential for healthy and hydrated skin. Conclusions. Human skin microbiota play a vital role in maintaining skin health and protecting against harmful microorganisms. While normal microbiota are generally beneficial, some microorganisms can be harmful under certain conditions, leading to skin infection or other health problems. Maintaining good hygiene practices, avoiding exposure to harmful microorganisms, and supporting the skin's natural defense system can help to promote skin health and prevent infections. By nurturing our skin natural microbiota, we can optimize skin health and contribute to our overall quality of life. That's all from me. Thank you, Doctor.